Very good afternoon to everyone. Welcome in today's class. So today we are going to talk about Krebs cycle. But before beginning with the topic, as we say, crack me. The topic is Krebs cycle. There are many other names for this cycle, talking about them also. But before moving with the topic, let us have a small introduction about Unacademy. So Unacademy is one of the India's largest platform for the I can see some of the students, they are live in the class, so they can please comment in the chat box that my voice is audible or not. Please reply me in the chat box that my voice is. So Unacademy is one of the India's largest platform for the preparation of NEET examination. One of the best educators all over India is found at Unacademy. So you can have this benefit of getting tutored by the best faculty all over India. And there are special classes. There are special classes, and what is so special about these special classes that you can ask your doubts and queries directly with the faculty member, and they're very much willing to solve your queries and doubts. So there are special classes. Then there are free tests and test series, and as I always say, it is very important to know the pattern of the examination and what kind of questions are going to come. So test series is an important part for the preparation. And that is also possible on Academy. There are free mock tests, weekly tests on Academy. Then this is one of the easiest way to prepare for the examination of an academy because there is flexible schedule. Then the language can be of your own choice. There are many faculty members who are teaching languages. Benefit of getting the lectures in your language as well. Unlimited practice. Once the video has been recorded, you can anytime go over there and watch it as many number of times as you want to. Then if it is about quizzes, then can be done as many number of times as you want to. So these are some of the important benefits of an academy. So what you need to do is you can just go to Play Store and write an academy learning app. So you'll be seeing the logo of an academy learning app. So just download this app and make your account, and you are good to go. You have access to all of the free classes, and you can prepare for the examination. Then you won't feel anything different than a normal classroom because you can chat with your educator, you can ask questions, and there are interactive posts that there will be questions um, asked to choose one of the options from those options, and each and every question will explain that why a particular option is the right answer and why other options are not right. So you can learn anytime, anywhere with the help of an academy. Then um, there is this plus subscription, and what is the benefit of this plus subscription? That all the 19 subjects are available at plus subscription, and for any upcoming exam there are short-term courses, crash courses, so you can get this benefit of always knowing that which exams are upcoming and what will be the pattern, and according to that the preparation will be going on at the platform, so you can also roll with it and prepare for the examination. So there are 12 monthly subscriptions, and it is for a very pocket friendly amount, and in this pocket friendly amount also you get an additional 10% discount by using my referral code FP1. By using my referral code FP1, you will be getting a discount of 10%, and the course will be much more reasonable. So this is it about the introduction part. Let us begin with the class, that is Krebs cycle. Krebs cycle. So the Krebs cycle is also called as T cycle. And what is the full form of TCA? That is tricarboxylic acid cycle. Tricarboxylic acid cycle. Now, okay, fine, we can understand why it is called as tricarboxylic acid cycle, because there are compounds which is having three carbon atoms, so it is tricarboxylic acid cycle. But why it is called as Krebs cycle, or why the name Krebs cycle is given, because this Krebs cycle was given by Sir Hans Krebs. Sir Hans Krebs. So this was the second cycle which was given by Sir Hans Krebs, and it was given in the year 1907. Then he got a Nobel Prize for the same in the year, Nobel Prize for giving the Krebs cycle in the year 1903, right? So this tricarboxylic acid cycle is also known as citric acid cycle, also called as citric acid cycle. Why? Because of the discoverer, that is Oxton, and it was in the year 1978 who described that tricarboxylic acid is the citric acid of tricarboxylic acid cycle. So it is also called as tricarboxylic acid cycle after the name of the scientist Oxton, and Oxton in year 1978, sorry, 1948 told that the tricarboxylic acid in TCA cycle is actually citric acid. So it is also called as citric acid cycle, tricarboxylic acid cycle, or Krebs cycle. And another name which is given for Krebs cycle is metabolic traffic cycle. Metabolic traffic cycle. This is the name given for tricarboxylic acid cycle, right? Why it is called as metabolic traffic cycle? Why is this word given, metabolic traffic cycle? Because there are intermediates in the Krebs cycle which are important in various other pathways. So intermediates it's in Krebs cycle is important various other metabolic pathways. Other metabolic pathways, right? Then moving ahead, what is the significance of this pathway? Significance of tricarboxylic acid cycle. So the first significance, or first of all, before beginning with the significance, the site of tricarboxylic cycle is mitochondria. Site is mitochondria. Now moving towards the significance, first one, that it is the final common oxidative pathway for carbohydrates, fats, and proteins conversion into carbon dioxide. So it is the final common, most importantly, common oxidative pathway for conversion of fats, carbs, and proteins to carbon dioxide. So whether it is fats, whether it is carbohydrates, hydrates, or whether it is proteins, each and everything will be finally uh, degrading into carbon dioxide through the last step, that is Krebs cycle. Second, it gives the reducing equivalents. It gives reducing equivalents. So what are this reducing equivalents? Those who are attending the class live can plan in the comment box. Yes, reducing equivalence. What is this reducing equivalence? What are these reducing equivalents? It gives NADH and DH. So these are the reducing equivalents given through the Krebs cycle. Now, what is the third significance of Krebs cycle? That it is helpful in synthesis of ATP. Then it is producing this ATP with the help of substrate level phosphorylation. ATP is given by substrate level phosphorylation, right? And by actual generation of GTP, which is finally giving rise to ATP. So generation of GTP occurs, and this GTP is converted into ATP. So it is responsible for substrate level phosphorylation, that is production of ATP without electrons transport chain. Production of ATP without, this is substrate level phosphorylation. So that also occurs with the help of Krebs cycle. So that was the third significance. Moving ahead, now oxaloacetate, which is formed in the reaction oxaloacetate, it acts as catalyst. In this pathway. It acts as catalyst in this pathway, and it's regulated in the pathway. So oxaloacetate acts as catalyst as well. And why is it called that it is a catalyst? Why is it called as catalyst? Because what is a catalyst? Whenever a catalyst is participating in a particular kind of reaction, what happens is this catalyst will remain as it is. So while the citric acid cycle, this oxaloacetate is actually not used up. This oxaloacetate is regenerated. So in the starting of the reaction, oxaloacetate is used up, and as it is called as cycle, that is a complete term, so it is regenerated. So the another term which is taken for oxaloacetate is that it acts as catalyst as well. So oxaloacetate is a catalyst. Then for the same, there is a very famous term in Krebs cycle, that is fat is burned on wick of carbohydrates. Carbohydrates. So what is the wick over here? In this case, what is the wick? Fat is burned, protein is burned, or carbohydrates, they are burned. They all are burned at a common wick. What is this wick actually? The wick is oxaloacetate. Oxaloacetate. So fat is burned at the wick of carbohydrates. So carbs, they are converted into fats, and fats can be converted into carb. So this kind of reaction, conversion of fat to carb and fat to carbs, this reaction is actually irreversible reaction. Particular reaction is irreversible. Irreversible reaction, and it is catalyzed by pyruvate dehydrogenase complex. So fat is burned on the wick of carbohydrate, and the wick is oxaloacetate. So this was the fourth significance of Krebs cycle. Forward towards the fifth significance of the cycle. This pathway is important for the synthesis of non-essential amino acid. This pathway important for synthesis of non-essential amino acids. So it is non-essential and the backbone of these amino acids, they are obtained from this pathway. So this is the thing. Then it is also synthesis of important in synthesis of heme, porphyrins, then purines and pyrimidines. So we will be seeing in the end of the lecture that from where the heme is formed from this pathway, how purines, how pyrimidines and how porphyrins are formed. Then coming to the next significance of Krebs cycle. So the intermediates which are formed in the Krebs cycle, or the various intermediates of various metabolic pathways, they come to the TCA cycle, and the intermediates of TCA cycle, they act as regulators in various other metabolic cycles or pathways. So we can say that intermediates of various metabolic pathways come from TCA cycle, and intermediates intermediates of TCA act as regulators in various other metabolic pathways. Yes, metabolic pathways. So please, those who are attending the class live, please they can do me a favor. They can just write in the chat box whether my voice is clear or it is breaking. So please, if you can do this much, it will be really a great help. So now talking about the steps of TCA cycle. Now, what are the steps of TCA cycle? So there are actually 8 important steps in the mitochondrial matrix which are related to tricarboxylic acid cycle. Clear, ma'am? Okay, thank you, Pragya, for your reply. Because I was like conscious. Yesterday my voice was like quite unclear. Thank you very much. So the steps of tricarboxylic acid cycle we are going to see. So there is a very great mnemonic to remember it. So there are 8 steps, and these 8 steps they occur in mitochondrial matrix. Mitochondrial matrix. 8 steps in mitochondrial matrix, and there is a very famous mnemonic. Remember the intermediates. So we'll see the mnemonic later on. First of all, let us move towards the steps. So first of all, oxaloacetate, the one which is called as catalyst of the pathway. Oxaloacetate. This oxaloacetate is converted into citrate. Is converted into citrate with the help of enzyme. So as I always say, if you can remember the name of the product, you can remember the name of the enzyme as well. The enzyme used over here is citrate synthase. Citrate synthase, right? And there is addition of acetyl CoA in this reaction. There will be addition of acetyl CoA. Acetyl CoA, and citrate is formed from this. Then this citrate is actually converted into cis acetonate, converted into cis acetonate, and this cis acetonate converted into isocitrate. Isocitrate. So the process of formation of citrate into cis acetonate is actually a kind of dehydration reaction. Dehydration. So by dehydration, cis acetonate is formed, and this cis acetonate is converted into isocitrate with the help of enzyme aconitase. And this process in which aconitase is participating is actually a rehydration reaction. dehydration reaction. So isocitrate is formed from citrate. This acetonate is like a very smaller intermediate. Then this isocitrate further converted into oxalosuccinate. 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 And this oxalosuccinate is further converted into alpha ketoglutarate. Alpha ketoglutarate. And the enzymes which are responsible for these reactions, this isocitrate is converted into oxalosuccinate, and the process of formation of oxalosuccinate from isocitrate actually a oxidative decarboxylation reaction. Oxidative decarboxylation. Carboxylation. So in this process there will be removal of carbon dioxide. Then this whole process, conversion of isocitrate into alpha ketoglutarate, actually what happens over here is there will be production of NADH, that is your high energy compound. So let us take another color and make a, there will be formation of NADH, NADH and positive. And this formation of isocitrate to alpha ketoglutarate, it itself is a two-step process. So there will be utilization of oxidative decarboxylation step, and there will be removal of carbon dioxide. And talking about the enzyme which is used here is isocitrate dehydrogenase. So the enzyme which is participating in this reaction is isocitrate dehydrogenase is the enzyme which is utilized over here, right? So now we are at alpha ketoglutarate. Fine. So alpha ketoglutarate is further converted into succinyl CoA. It is converted into succinyl CoA. So in this process, the enzyme which is used is alpha ketoglutarate dehydrogenase, and again this step yields energy, that is there is conversion of NADH plus H positive ion, and this step is also a kind of oxidative decarboxylation like here we have seen. So here also there will be removal of carbon dioxide. This is the oxidative decarboxylation reaction. So there will be removal of carbon dioxide, right? So here there will be formation of one carbon dioxide, and here also I actually just rubbed it, so I need to write there will be removal of carbon dioxide at this step also. So here this I am writing that formation of NADH occurs from NAD. So I have highlighted it over here. So this formation of NADH and carbon dioxide is actually a process where isocitrate is converted into alpha ketoglutarate, and oxaloacetate is just a side product. Right now we have this succinyl CoA. Now this succinyl CoA is changed into succinate. Changed into succinate. Now, this succinate, it is formed by the enzyme succinate thiokinase. Formation of formation of succinate from succinyl CoA is actually dependent on succinate thiokinase. Succinate thiokinase, right? And this step is actually substrate level phosphorylation, where there is no need of electron transport chain and there is formation of ATP. So this step gives ATP. ATP, or it is in the form of GDP, which is later on converted into ATP. So there is formation of GTP. So this is a substrate level phosphorylation step. That is the product. ATP is formed without any kind of electron transport chain. Very important to remember. So now your succinate is formed, right? Now this succinate is going to change into change into fumarate, and this step is also high energy yielding because there will be formation of FADH2 from FAD. So this step is also energy yielding, and the enzyme which is responsible for this kind of reaction is succinate dehydrogenase. The enzyme used over here is succinate dehydrogenase. Dehydrogenase. Right? Now this fumarate is converted into malate. Fumarate is converted into malate, and this malate is converted into oxaloacetate. So the conversion of fumarate into malate, there is requirement of hydrogen, and this step is actually catalyzed by enzyme fumarase. And this malate is converted into oxaloacetate. This is again energy yielding step, that is there will be formation of NADH from NAD. So this is how the whole Krebs cycle looks like. Let us talk about it in a very different manner. That is, this oxaloacetate is actually a 4-carbon compound. A 4-carbon compound. Acetyl-CoA is a 2-carbon compound. Let us just put these things in highlight like this. So 4-carbon compound and 2-carbon compound, they both join to form a 6-carbon compound, that is citrate. The citrate is a 6-carbon compound, right? Now the process of formation of isocitrate from citrate. So the name itself is telling that iso. So it's a kind of, this reaction is a kind of isomerization kind of reaction, 2-step process, and it is the isomerization reaction, that is formation of isocitrate from citrate, and the process is catalyzed by aconitase, and it is a kind of dehydration reaction when there is formation of cis aconitate from citrate, and when cis aconitate is converting into isocitrate, the enzyme utilized is the process is actually rehydration. When this isocitrate is getting converted into alpha ketoglutarate, there will be formation of NADH, which is very important, at which places the energy is generated. So NADH is generated in the process of formation of alpha ketoglutarate from isocitrate. Then second energy is generated when the formation of alpha ketoglutarate into succinyl-CoA, and there will be production of NADH. Now succinyl-CoA will be changing into succinate. This is again energy yielding step. And succinate is changing into fumarate. It is again energy yielding step. So back to back there are 4 energy yielding steps. Then formation of malate from fumarate is non-energy producing state. Step, and formation of oxaloacetate from malate gives energy. So there are 5 places in Krebs cycle which are actually yielding us the energy. So we will be also talking about energetics of Krebs cycle, but please try to remember what are those places where energy is generated. This is a very important thing. So how many steps have you seen till now in Krebs cycle? How many steps we have seen? It is made up of majorly 8 steps. The major 8 steps is oxaloacetate getting converted into citrate, citrate getting converted into isocitrate, isocitrate getting converted into alpha ketoglutarate, alpha ketoglutarate getting converted into succinyl-CoA, succinyl-CoA getting converted into succinate, succinate getting converted into fumarate, fumarate getting converted into malate, and again there will be generation of oxaloacetate, which is the catalyst of the whole process because it is regenerated. So you can count the 8 steps: 1 oxaloacetate, 2 citrate, 3 isocitrate, 4 alpha ketoglutarate, 5th succinyl-CoA, 6 succinate, 7 fumarate, and 8 malate. Right? So this is the whole Krebs cycle. So we are just done with the Krebs cycle, but not with the today's lecture. We have to read many more things. So the mnemonic which I was telling you at the starting of the class, that I'll be telling you about the mnemonic also. So what is the mnemonic? The mnemonic is citrate is Krebs is starting starting substance for making oxaloacetate. So you can remember it like this. The first citrate is Krebs starting substance for making oxaloacetate. So remember it this way. C over here is for your citrate, I is for your isocitrate, Krebs is for your alpha keto glutarate, S is for your succinyl CoA, then S is for succinate, F is for fumarate, M is for malate, and O is for oxaloacetate. So citrate is Krebs starting substance for making oxaloacetate, right? You can remember this much. So moving back to the cycle, the important thing which I wanted to say about this, that the two carbon compound, that is acetyl CoA, is removed at the, so this two carbon of acetyl CoA are removed at the successive steps. So we have also seen where they are actually removed. So let us take another color. Yeah. So one of the carbon dioxide is removed over here and one of the carbon dioxide is removed over here. So also remember that in formation of oxaloacetate from isocitrate, there is formation of, or there is removal of carbon dioxide, or can say that in formation alpha ketoglutarate from isocitrate, there is removal of carbon dioxide, and in formation of succinyl CoA from alpha ketoglutarate, there will be removal of carbon dioxide. So these are the steps. So the two carbons which were actually added from acetyl CoA are removed in the cycle itself, right? So let us move ahead and talk about the further things. So let us talk about the energetics of the reaction. Energetics. So there is production of 3 NADH and there is production of 1 FADH. We'll go back to the cycle also for seeing at which places they are done. So we have to see 3 NADH, right? So 1 NADH here, 1 NADH over here, and 1 NADH over here. I hope you people are seeing where I'm moving my pointer. So laser point. See, 1, 2, and 3. These are the three places where NADH is produced. This is the place where FADH is produced, and this is the place where GTP is produced, and which is in the end giving rise to ATP, right? So moving ahead, in the energetics, 3 NADH will give rise to 7.5 ATP, and 1 FADH2 will give rise to 1.5 ATP, and ATP is ATP itself. So the total ATP which is generated in Krebs cycle is 10 ATP, 10 ATP, right? So in one turn of acetyl CoA, we are getting 10 ATP. So we can say per turn of acetyl CoA, we get 10 ATP. Per turn of acetyl CoA, we get 10 ATP. So for one glucose, we are going to get 20 ATP. But in one Krebs cycle, we are going to get 10 ATP. Understanding that what I am trying to say? 1 glucose will give you 20 ATP in Krebs cycle. ATP in Krebs cycle. So only for Krebs cycle, not for glycolysis. This 20 ATP is only for Krebs cycle. And one cycle of Krebs cycle is giving you 10 ATP. Right? So this is about energetics. Now, another important topic which we need to know over here, the another thing that we need to know over here is about the inhibitors of TCA cycle. Inhibitors of TCA cycle. Inhibitors of cycle. So I'll be telling you about the name of the inhibitors and the enzyme which it is inhibiting. So the first inhibitor that you need to know is fluoroacetate. Fluoroacetate. So fluoroacetate will give rise to conitase. Then arsenite will give rise to, acetate is inhibiting aconitase, and ketoglutarate is getting inhibited by arsenate. Then malonate will inhibit succinate dehydrogenase. Malonate will inhibit succinate dehydrogenase. So this we have talked about the inhibitors of TCA cycle. Fluoroacetate is going to inhibit aconitase, arsenite is going to inhibit alpha ketoglutarate dehydrogenase, and malonate is going to inhibit succinate dehydrogenase. Right? Now talking about the regulation, which is also quite important, that how the Krebs cycle is regulated. So talking about the regulation of Krebs cycle. So that Krebs cycle, it is regulated on the basis of availability of substrate. The very first way by which it is regulated is on the availability of substrate. First thing. Second thing, that ATP is a very, very important regulator of Krebs cycle. How? So ATP is important regulator. How is it regulating the Krebs cycle? So what will it do? It will stimulate the pathway. ADP is going to stimulate the pathway. The third way through which it is regulated is by 3 enzymes, that is citrate synthase, then isocitrate dehydrogenase, then alpha ketoglutarate dehydrogenase. So these are the 3 enzymes which are actually allosterically regulated. These 3 enzymes are allosterically regulated. So first, it is dependent on the availability of substrate. Secondly, ADP is important regulator. Then these 3 enzymes are allosterically regulated. Right? So those who are attending the class live, they can just write in the chat box, are they understanding what I am trying to say? So please write in the chat box whether you are understanding clearly. So next, ATP. Whenever there will be excess of ATP, it will inhibit the pathway. ATP inhibits pathway. And ADP will stimulate the pathway. So the role of ATP is different than role of ADP. Because when there will be lack of energy, the cell will try to know that there is lack of ATP as there is lots of ATP, so it will just shift the cycle into the positive direction. Then if there is again in energy in the form of NADH, it will also inhibit the pathway. It will also inhibit pathway because there is lots of energy. Then sixth point, or the sixth way how it is regulated is with the help of succinyl CoA. Succinyl CoA. So the succinyl CoA actually inhibits alpha ketoglutarate dehydrogenase. So we just saw that these three enzymes, citrate synthase, isocitrate dehydrogenase, and alpha keto dehydrogenase, these three enzymes are allosterically regulated. So succinyl CoA, what is it going to do? It is going to inhibit alpha ketoglutarate. Inhibits alpha ketoglutarate dehydrogenase enzyme, right? Then moving to the next, that acetyl CoA. Acetyl CoA. Acetyl CoA, it inhibits pyruvate dehydrogenase. Inhibits pyruvate dehydrogenase. So these are 7 ways through which the TCA cycle is regulated. So let us have a very quick revision of the way it is regulated. So on the basis of availability of substrate, ADP is positive regulator. Then these three enzymes, citrate synthase, then isocitrate dehydrogenase and alpha ketoglutarate, they are allosterically regulated. Then ATP inhibits the pathway, NADH inhibits the pathway, succinyl CoA, it inhibits the enzyme alpha ketoglutarate dehydrogenase, and acetyl CoA, it inhibits the pyruvate dehydrogenase, right? So this is about the regulation of Krebs cycle. Now talking about amphibolic role of tricarboxylic acid cycle. Amphibolic role. Amphibolic role of a cycle. So what do we mean by this amphibolic role of TC cycle? What does this mean actually? So this cycle, this TCA cycle, it acts as a anabolic pathway as well as catabolic pathway. So working as a catabolic pathway and working as a anabolic pathway makes it a amphibolic pathway. So it has got amphibolic roles. And now we are going to highlight, or we are going to read the amphibolic roles of tricarboxylic acid cycle, right? So first of all, we'll write that this cycle is anabolic as well as catabolic. Thus, it's amphibolic. Thus, it's amphibolic, right? So now see how amphibolic it is, or where all the intermediates acts as amphibolic. So first of all, talking about acetyl CoA. So acetyl CoA is converted catabolically into carbon dioxide. So here we can write catabolically. Catabolically is converted into carbon dioxide. So all the foodstuffs are actually oxidized into carbon dioxide by this cycle. So all all stuffs are oxidized into carbon dioxide by a cycle. So this was the first thing. So this is the catabolic role we have seen. So second role we are going to see is anabolic role. Anabolic role. Anabolic role. What are the anabolic rule? The very first anabolic rule is formation of aspartate from oxaloacetate. Oxaloacetate will give rise to aspartate. Aspartate. And this aspartate is very much important in purine and pyrimidine synthesis. Important in purine and pyrimidine synthesis. Right? So the first anabolic role. Second anabolic role we are going to see. So let us just write over here. 1. Second anabolic rule is with the help of citrate. Let's write citrate. So citrate is important source of acetyl-CoA. Important Citrate is important source of acetyl-CoA. So important source of acetyl-CoA, where exactly where does it give rise to this acetyl-CoA? It's important source of acetyl-CoA in cytosol. Remember this, in cytosol. So it is important source of acetyl-CoA, and acetyl-CoA itself is required for. Acetyl-CoA is required for what all things? We have done this in many other places also. It is required for fatty acid synthesis, and it is required for cholesterol synthesis. Cholesterol synthesis. So this was the second anabolic role, that the citrate is important source of acetyl-CoA in cytosol, and acetyl-CoA actually is required for synthesis of fatty acids and cholesterol. Now moving ahead, talking about the third anabolic role of Krebs cycle. So this alpha ketoglutarate required for formation of glutamate and GABA. So alpha ketoglutarate is required for synthesis of non-essential amino acid. Alpha ketoglutarate is required for synthesis of non-essential amino acid. Now talking about the fourth anabolic role is because of succinyl-CoA. Our succinyl-CoA is required for heme synthesis. So the very first reaction, which is the condensation reaction, in that reaction succinyl-CoA is used up. So succinyl-CoA is required for synthesis of heme. And it is also important for ketolysis. So succinyl-CoA, alpha ketoglutarate, then citrate and oxaloacetate. These all things, they play important role in anabolic pathway. Oxaloacetate for aspartate, then citrate for acetyl-CoA, which itself is required for fatty acid and cholesterol synthesis. Then alpha ketoglutarate is required for glutamate, and succinyl-CoA is required for synthesis of heme. Right? So we have seen its amphibolic role. Now talking lastly about anapleurotic reactions. So anapleurotic reactions. So which reactions are called as anapleurotic reaction? Which reactions will be actually falling in the category of anapleurotic reactions? So the reactions which replenishes the intermediates of TCA cycle are called as anapleurotic reactions. So reactions, let us write that also, reactions which replenishes the intermediates intermediates of TCA cycle called as or is known as anapleurotic reactions. So let us talk about the anapleurotic reaction. So we'll be talking about 7 anapleurotic reactions individually, right? So the first anapleurotic reaction, the first one is formation of oxaloacetate from pyruvate. Pyruvate gets converted into oxaloacetate. So this is the anapleurotic reaction. So the enzyme which is utilized over here is pyruvate carboxylase. Pyruvate, pyruvate carboxylase. Pyruvate carboxylase is the enzyme which converts pyruvate into oxaloacetate. And whenever there is a carboxylation reaction, it is dependent on ATP, it is dependent on biotin, and it requires carbon dioxide. So ATP, biotin and carbon dioxide, which is always required for carboxylation reaction, also required over here. So this is the first anapleurotic reaction. Then second anapleurotic reaction is glutamate. Glutamate changes into alpha ketoglutarate. Changes into alpha ketoglutarate. And the enzyme which is responsible for this reaction is transamination and deamination enzymes. So the reaction is transamination and deamination. Transamination and deamination reactions for formation of alpha ketoglutarate from glutamate. This was the second anapleurotic reaction. Talking about the third anapleurotic reaction is the change of arginine or arginine, proline and histidine getting converted into alpha ketoglutarate. Alpha glutarate. So from arginine, proline and histidine, alpha glutarate is obtained. Third anapleurotic reaction. Now fourth anapleurotic reaction is from propionyl CoA. Propionyl CoA. So this propionyl CoA will change into succinyl CoA. Will change into succinyl CoA. And this propionyl CoA actually obtained from valine, then methionine, then isoleucine, then threonine. Then threonine, valine, methionine, isoleucine, and threonine. Propionyl CoA will get converted into succinyl CoA. So this was the fourth anapleurotic reaction. Now the fifth anapleurotic reaction is conversion of phenylalanine and tyrosine, and also from urea cycle. There will be formation of fumarate. I have seen the urea cycle as well. So if you are having any kind of doubt in this, you can go and check my video on urea cycle. So formation of fumarate from phenylalanine, from tyrosine and from urea cycle. Then the next anapleurotic place is formation of malate from pyruvate. Pyruvate will change into malate. This is also the anapleurotic reaction, and the enzyme which is used over here is malic enzyme. Malic enzyme. And this malic enzyme is actually NADPH dependent. NADPH dependent. So this reaction is NADPH dependent reaction. The formation of malate from pyruvate is anapleurotic reaction. Then last is formation of oxaloacetate from aspartate. Aspartate. Formation of oxaloacetate from aspartate. So the formation of oxaloacetate from aspartate is actually a transamination reaction. Transamination reaction. So we have seen 7 anapleurotic reactions where there is regeneration of the intermediate of the tricarboxylic acid cycle. So there is formation of oxalate from pyruvate. There is formation of alpha ketoglutarate from glutamate. There is formation of alpha ketoglutarate from three amino acids, that is arginine, proline and histidine. Then there is formation of succinyl-CoA from propionyl CoA. Then there is formation of fumarate from phenylalanine, tyrosine and urea cycle. Then formation of malate from pyruvate, formation of oxaloacetate from aspartate. So like this we have covered the Krebs cycle in detail, and let us have a very quick revision. So it is also called as tricarboxylic acid cycle. It was given by Sir Hans Krebs in the year 1937. It is also called as metabolic traffic cycle. So why it is called so? Because intermediates of Krebs cycles are important in other metabolic pathways as well. So significance, we have seen that this is the common pathway for the production of for the oxidation of fats, carbohydrates and proteins, and it gives the reducing equivalence of NADH and FADH2. There is synthesis of ATP. Then oxaloacetate acts as catalyst. Then there is a very famous saying that fat is burned on the wick of carbohydrates, and that wick is oxaloacetate. Then the pathway of citric acid cycle is responsible for non-essential amino acid synthesis, important in heme, porphyrins, purines, and pyrimidine synthesis. Then we have seen the whole cycle in very detail. So there is a mnemonic citrate is Krebs starting substance for making oxaloacetate. You can remember this, and you will remember the all intermediate sequence. And in one Krebs cycle, there is formation of 10 ATP, and one glucose will give 20 ATP with the help of Krebs cycle. Then we have seen the inhibitors of TCA cycle. Fluoroacetate, it inhibits aconitase. Arsenite, it inhibits ketoglutarate dehydrogenase. Malonate, it inhibits succinate dehydrogenase. Then we have seen the regulation in detail, that how it is regulated. Then we have seen the amphibolic role of TCA. Then we have seen the anapleurotic role of tricarboxylic acid cycle. So for this we have seen 7 places where the intermediates of tricarboxylic acid cycle are produced. So like this we have covered almost everything in TCA cycle. So that is it for today's class. And lastly, please subscribe our channel, that is Let's Crack NEET PG, and hit the bell icon for getting all the notifications of the upcoming classes. And for this plus subscription, use my referral code, that is FB01, and you will be getting 10% discount in this plus subscription. Also, do not forget to go and watch my videos on other topics of biochemistry. There is immense lectures which I have made on biochemistry, and you will be getting these lectures on Let's Crack NEET PG channel. So just go and check out my other videos as well, and please give me a thumbs up. It really motivates us when we see your thumbs up and your likes. It really motivates us. And this is it for today's class. We'll be meeting again in another class with some other interesting topic of biochemistry. So till then, be happy, keep on enjoying, and this is it for today's class. Bye.